Biomedical Engineering, being named a Woman of Innovation in 2017, a Young Innovator in Cellular and Molecular Bioengineering in 2015, and served as the President of the Canadian Biomaterials Society in 2017 to 2018. Today, Dr. Willerth will provide us some valuable information on the basis of research. Questions related to this webinar will be conducted in the end of the session. If you have any question, you can raise your hand and write in the comment section. Without further ado, I would like to request Dr. Willerth to please proceed with today's session. Dr. Willerth, over to you.
All right. Let me find my slides. Why not? Okay, 1 second. Okay, there we go. Perfect. Hopefully everyone can see my slides. Thanks again for this invitation. It's always interesting to work with groups across the world. And today I'll talk a bit about the work we've been doing in 3D printing complex tissue models. And let me flip these so it looks normal, I think. Just duplicate slideshow. Okay. Is that working? Can you see my slides?
Yes, the slides are visible.
Okay, perfect. All right, let me find that. Okay. Yeah, so I'm located in the University of Victoria. So we are off the west coast of Canada, north of Seattle and west of Vancouver. And our lab has been really lucky to work with a lot of different bioprinting technologies over the years. Also, I can't see any hands raised, so just speak up if you have a question. I do have a company called Biosciences that does sell bioinks, and I am the CEO and co-founder of it. Yeah, so I know a lot of you are biologists, and I think one of the fields that's really exciting is the field I work in of tissue engineering, where we apply the principles of biology and engineering to develop functional substitutes for disease and damaged tissue. Some of the classical examples are Wake Forest, where they've made bladders that can go into people, tracheas. Vacanti famously grew an ear, a human ear on a mouse. And something that was really, really exciting that just came out in the last few months is a company called 3D Biotherapeutics out of New York. They actually took a patient's own stem cells and 3D printed them into an ear shape and used this to treat deformities in ears. And so this is the first time that 3D bioprinted tissues have been transplanted into people. And I think it's a really big deal for the field and the potential for this technology. Neural tissue engineering, lots of opportunities there. Probably most of you know someone who's had Alzheimer's or Parkinson's disease. I'm a really big race car fan, and Michael Schumacher suffered one of these horrible traumatic brain injuries, and even with all of his resources and being like the wealthiest F1 driver of all time, still hasn't really made much progress. And so neural tissue engineering allows us to understand these diseases and hopefully identify better treatments. And a lot of our 3D bioprinting work, I always say we're not going to 3D print a brain that you'd put back into a human, but it can be used to generate these human models of drug discovery and development, specifically Alzheimer's, which I'll talk a little bit about today, and Parkinson's as well. I'm just showing one of the recent drugs from Biogen, Abdulham, which was approved, but was still super controversial, as essentially they spent so much money, the drug had to be approved, and doctors really don't like prescribing it. So there needs to be a better way to do this process besides using animals, which is the main method that people currently use. And they tend to be, for all the graduate students out there who work with animals, they tend to take a lot of work. As a professor, they tend to be expensive, and then they lack predictive capability. There is always the possibility to get human tissue slices cultured in vitro, but they tend to be limited. So what our group has done has actually been looking at how we can make neural tissue from stem cells, and in particular, human-induced pluripotent stem cells. So back in 2006, Takahashi and Yamanaka, wow, and it's coming up on 12 years since they won the Nobel, showed you could take a stem cell, treat it with these factors, and they would go back to a stem cell-like state where they could become any cell type found in the body. The trick is trying to get them to become neural tissue. And I wrote a book on this during my sabbatical. back in 2016 on engineering neural stem cells. And I'm actually currently writing my second book on the topic of neural engineering for LSIVA as well. So you have these cells that can become any cell type. We wanted to make the cells found in the central nervous system, which include your neurons, which are the excitable cells that transmit information using electrochemical signaling. Oligodendrocytes help insulate the neurons by covering them with a myelin sheath and helping the signal propagate. And then the astrocytes are these support cells, which provide nutrients to the neurons and oligodendrocytes. And they also form the blood-brain barrier. So your brain and spinal cord are actually immunoprivileged and protected from the rest of the body by this thick layer of astrocytes. And while I'm not going to talk about it today, there is some really nice work being done in the field to model this blood-brain barrier. And that's one of the reasons it's so hard to treat nervous system disorders, is getting across this barrier. The way we get our stem cells to become our neural tissue is we actually rely a lot on small molecules. And here are some of the ones we use a lot in my lab. We like them because they're similar to growth factors, but tend to be more affordable. You can screen libraries of these small molecules, and they tend to be less sensitive to conditions like temperature and pH. The first one that we use is a canonical one called retinoic acid, which tells stem cells to differentiate into neural lineages. And during development, this vitamin A derivative is what establishes polarity in your spinal cord, forming your hindbrain and the neural tube. Pure morphine acts similar to a protein found in our body called Sonic Hedgehog, which was named after the video game by the Japanese people who discovered it. And in fact, I used to have, and I've been a prof for over 10 years, I always used OG Sonic, and now he's a movie. I guess they've made 2 movies about Sonic. And what's interesting about this protein and the small molecule that acts like the protein is that patterns the ventral neural tube. And so this specifically will differentiate our neurons into motor neurons, which are those neurons responsible for motion. And this is important when we try to model the spinal cord and injuries to the spinal cord. And the final drug we use a lot is glucosterone, which induces the differentiation of our iPSCs into dopaminergic neurons, which are the cells that die off during Parkinson's disease. Interestingly, glucosterone is a plant steroid that's normally used as an anti-cancer drug, but a large-scale screen done by Gonzalez et al. Found it to be the most effective inducer of dopaminergic differentiation when they screened 1,000 small molecules. And later, my group showed similar properties when it was used to treat induced pluripotent stem cells. Okay, so why did our lab get into bioprinting? So there's lots of different ways to engineer neural tissue out there. And in fact, this picture right here is a brain organoid, which is a technique our lab has also used. But differentiating these cells into mature neural tissues takes weeks and often lots of cell culture media changes. And for those of you who know cell culture, you know that, yeah. So if you want to do a high throughput version, it's really hard. It's really going to be dependent on you changing the media on a bunch of different cultures. When you rely on stem cells to self-organize, like these organoids, you can sometimes get structures that extend out in all directions, not accurately replicating the structures found in vivo. And one of the reasons pharmaceutical companies haven't really adopted organoids is because they experience variability from culture to culture. And so 3D bioprinting can address these limitations. So what is 3D bioprinting? Weirdly, I'm at my home, but I don't have any of my 3D printed animals with me. Because usually I pick that up and I say 3D bioprinting is similar to traditional 3D printing, but instead of extruding out a filament, we use specialized bioinks that contain these cells to produce the structures. And that's where my company Axolotl comes in, is that it has these really good biomaterials for printing stem cells as well. And then you can print them into these tissues and culture them and use them for drug discovery. And as I said, I'm not too optimistic about them being used for transplanting regions of brain. As I mentioned, the ear application is a really good one, and there's also some artificial pancreas work as well. And if you want to read more, I collaborate a lot with a company called Aspect Biosystems, who makes one of the high-end bioprinters we use, and this came out in Current Opinion in Biomedical Engineering back in 2020. So if you're thinking about bioprinting, here are some things to consider. One is the method of printing, and while I'll be talking about a rather expensive bioprinter, there are cheaper ones you can make, and in fact, there are protocols out there to convert normal 3D printers into bioprinters for around like $1,000. The choice of cells you want to use, the number, quantity. One of the reasons our biology is so good is we actually print at around 1 million cells per mil, so we are really replicating the numbers of cells you would see in a tissue found in the human body. The choice of bioink, you're usually looking at a trade-off between printability and keeping your cells alive. What structure should you print? Can that structure be printed? And can you incorporate growth and differentiation factors into the tissues? As I mentioned, we collaborate a lot with Aspect Biosystems, and they have, as you see here, a very fancy printer called the RX1. And then they have these microfluidic print heads. So for those of you who've done microfluidics, essentially it has, a standard one has 4 inputs, so you can switch between 2 materials or 2 cell types, and also run a cross-linker solution through. They also have some that will print coaxial, which we'll talk about in a little bit as well. And this is, I think, just showing how the print heads work and how you have the different inputs and they mix inside of the print head. And you have to sort of carefully time them so when it extrudes that your fiber being printed actually comes out solid. As I mentioned, I've collaborated a lot with Aspect Biosystems. So we had initially started our partnership back in 2016, and we've successfully leveraged that into a number of different funding programs. One of my graduate student and co-founder of Axolotl, Dr. Laura De La Vega, actually went and worked at Aspect as part of her MyTex internship. And we've written several papers together with authors from both academia and industry. And we actually received the top university award for excellence research partnerships back in 2020. And this is a photo of me and Conrad, who is a professor at UBC and one of the co-founders of Aspect, at an awards gala pre-COVID when we won one of our awards that came with a giant check. So I always like to show that there because giant checks are great. And I also like to point out that like you don't have to, like it's not exclusive to just be in academia or in industry, and there's really good ways to collaborate with both. As I mentioned, in terms of our bioink, our lab has spent a lot of time and effort developing a printable version of fibrin called tissue print, which is currently available on our website, axolotl.Biosciences.com. We published this in, I think, 8 or 9 peer-reviewed journal papers, and it was actually the initial paper detailing tissue print on the Aspect system was selected as a method of the year for ACS Biomaterial Science and Engineering. And if you are looking to get more into bioprinting, I'll really highly recommend checking out our STAR Protocols paper, which also is the first paper to detail how to use tissue print to print human tissue models. But it also has a cheaper version of the bioink made from alginate and gelatin that's very affordable. So if you're looking to try this out on potentially a homemade bioprinter, I would look at that as well, just to get you some cheap bioinks that you can use to start printing and seeing how the printing process works. Also, if you want to learn more about our natural bioinks, Claire, who just successfully defended her thesis on bioprinting Alzheimer's models, Josie, who was our top BME student, Becca, who's defending soon, Nadia, my lab manager, and Hannah and Kira, 2 more of our BME students, wrote this wonderful review on natural biomaterials that I think has already been cited like 45 times since it came out a year ago. So I think it gives a really good overview of all the different materials out there. As I say, there's always no perfect bioprinter and there's no perfect bioink. And so this can give you an idea of what potentially can be used. Another paper that also came out from our group, Zaina and my lab manager, Nadia, they wrote a review paper about the use of smart bioinks that respond to stimulus. And this again also came out. And both of these are open access, so they're freely available for everyone to read. So with this concept of smart bioinks, we incorporate into our bioinks these drug-releasing microspheres that release those morphogens I talked about earlier. So we have these small spherical particles fabricated from biocompatible polymers, and they can provide tunable drug release. And we'd initially developed these as a tool to promote stem cell differentiation, but once we got our bioprinter, we then were like, oh, we can just add these to our inks and sort of cut down on the amount of work needed to be done. And here's just some of our previous work showing we can get controlled delivery of glucosterone, which I previously mentioned, over 45 days using these microspheres. Laura did similar work over another extended time period for the pure morphamine as well. And we currently have the same work being tested by Maria, who's in my lab, who received an internal research award called the J. Cure Award, who's looking to see how the release is effective when you actually have these suspended in bioink as opposed to just in the microsphere form. So we actually did add some of these microspheres to our bioink, and Ruchi Sharma, who defended her PhD this past April, she published a paper showing the physical and mechanical characterization of our fibrin-based bioprinted constructs. And she did this to show that when we added the microspheres, it made them stiffer, but more similar to brain tissue in this case. And her paper was selected out of 177 to be on the cover of the journal Processes. Interestingly, with our RX1, we can actually precisely place multiple different types of microspheres inside of our bioprinted tissues, which gets us towards patterning, which is quite exciting here. So in this case, one of the things I'll talk about is when you normally print cells or you print microspheres, they all look the same under a microscope. They look like these little dots. And so we found in order to show we're getting things localized where we want them to, we'll actually include these fluorescently labeled microspheres so we know where in the tissue different things are going, and in terms of cells, as well as drug-releasing microspheres. So as I mentioned, Dr. Laura de la Vega, who's one of the co-founders of Axolotl and was one of my awesome PhD students, she had this great paper come out where she used 3D bioprinting of human-induced pluripotent stem cells and drug-releasing microspheres to produce responsive neural tissues. And this came out in Advanced Nanobiomed in 2021. Again, open access, so anyone can read it. So here she's attempting to print miniature spinal cords. You can see them here. They're these little cylinders. The little red dots you see are actually the fibers that were extruded to make these constructs. We had 4 different groups. This negative control only contained stem cells. We had a bioprinted, again, neural progenitors, but in this case, we added the small molecules to the medium. We also had a group with unloaded microspheres, as we find that the microspheres, even without drugs, will promote more stem cell proliferation. And then we had the loaded microsphere group. Laura did a lot of nice work to show we get really even distribution of our microspheres of both kinds of drug releasing and the stem cells throughout the constructs. We then cultured them for 30 days. So one of the really nice things about our ink and its formulation is that it allows you to culture these constructs for up to 60 days for some of our Alzheimer's work. And we found that we're producing really relatively mature tissues thanks to our drug-releasing microspheres. So here, all the tissues contained astrocytes, all of them contain neurons, and you can see in the loaded microsphere group, we see more neurons. We also stained for chat, which interestingly, of course, in the positive control and the loaded microsphere group, we saw a lot more chat staining, which is good because we wanted to produce motor neurons like those found in the spinal cord. Also, I think this was the first time anyone had bioprinted tissues where we did see expression of myelin. So we were getting relatively mature oligodendrocytes, and our tissues also contain GABA, which is a neurotransmitter. So again, seeing a really high level of maturity in our tissues. And the final thing that was really, really exciting about these is we used a voltage-sensitive dye to measure our tissues at rest and after neurotransmitter stimulation. And we found that they were able to respond to neurotransmitters. So we actually produced a functional neural tissue. So now we can screen these tissues for 2 different things for drugs. So you can look at both, is it going to kill the cells? And 2, is it going to affect the neural activity of the tissues as well? So we were really excited to see these differences in membrane potential. So the conclusions from this study were the microsphere-containing bioinks promoted the development of functional neural tissues. Using controlled release of PRNRA from the microspheres effectively promoted the differentiation of bioprinted stem cell tissues. And interestingly, compared to some of the 2D culture models, these tissues actually are not only 3-dimensional, but they have all the cell types found in the spinal cord, which makes it a much more physiologically relevant model if you're going to be looking at drug screening. And just to talk a little bit about how this connects to the company, we actually got follow-up funding through the Next Generation Manufacturing Supercluster in Canada and Innovate BC to work with a local company, Starfish, to actually scale up the process of how we make those drug-releasing particles. So we recently had a paper come out about generating these therapeutically relevant microparticles with Alejandro and Arash from Dr. Catherine Elvira's group and Rebecca Kirsch from my group. And Starfish Medical is actually, it's been really interesting to see how we go from a PDMS device to some of their commercial grade microfluidic devices. And we're in fact presenting on that this Thursday. But yeah, if you're interested, you can check out the paper. Again, another great industry academic collaboration. And Catherine's great. And here's the paper, so you can again see, we used to make these in batches where you would shake them up. And then Alejandro's been able to make them in a more consistent fashion using microfluidics. And this is just talking about the different parameters there. One of the other things that I've been really excited about, John Walter Shumka, who's been in my group for a while now, he's been working on a collaboration with Dr. Hakan Nygaard, who runs the Alzheimer's Clinic over at the University of British Columbia. And so he actually takes blood draws from his patients and then uses this iPSC reprogramming process to make cell lines And we've been able to bioprint these into neural tissue that express these symptoms of Alzheimer's. And we hope to use these as a tool for drug screening. So this is just showing in 2D culture, you see increases in tau and amyloid beta, which is what you would also see in vivo in these patients. So you can see as the tissues age, they tend to get more Alzheimer's over time. This photo is just from some of John's tissues. I always include it because this was the last image we took before my lab had to shut down for good due to COVID. And so John got permission to take the cultures to 45 days, and then we had to shut down all of our tissue culture and everything in lab. And then when he went back 3 months later, he went and stained them and found that they did express neuronal markers and neurites at day 45, and we got some really lovely images. You can see because they were Alzheimer's, the neurite extinction isn't as pretty as they are with healthy cells. Okay, so with that, I'm going to talk a little bit more about a different model and switch gears to talk about glioblastoma or cancer. So glioblastoma is a deadly primary central nervous system tumor that originates from the glial cells, such as astrocytes, and exhibits invasive proliferation. It's a horrible disease. One of my friend's fiancés had it, and it was horrible. You essentially undergo chemo. They'll do surgery to remove the tumor, but it almost universally recurs and is almost universally a death sentence. So Chris Lee, who actually did his master's and is currently doing his MD PhD under my supervision with Dr. Nygaard, when he joined our lab, he was really, oh yeah. What's up? Okay. Let me know if you need anything. Yeah, so when Chris joined my lab, he was really interested in doing cancer, which our lab hadn't done at that point. And since we had the bioprinter and the bioink, he decided to go ahead and get some human glioblastoma cells and print them into rings. And the reason we used rings is because that's one of the default structures that came on the system. And as you can see, the cancer cells really liked it. They had really high levels of viability in our ink. They also formed tumors. So we saw these tumor spheroids that would spontaneously form inside of these 3D constructs, which makes sense because this is what they would do in an in vivo setting. Interestingly, when you compare them to 2D cancer models, they had increased CD133 expression and decreased TUJ1 expression. So essentially, by culturing human cancer cells in 3D, they acted more like a cancer tumor would. And this, one of the reasons for doing this is that a lot of drugs are screened in 2D, and it's not really replicating the same structures that you'd find in a human body. So. And previously, before Chris had moved into 3D printing, he had done a really nice study where he showed that, again, these small molecules, this combination of 5 small molecules would take a tumor cell that's highly proliferative and turn them into these non-differentiating, non-proliferating neurons. So he was really excited to see what would happen when we use this in our 3D system. And it turned out it wasn't as effective, probably due to the fact that it's much harder for those drugs to get into the spheroid than it is when you just treat things in 2D culture. And so in this case, he found that the cocktail slowed proliferation but did not inhibit tumor formation, which was a little bit disappointing, but it also makes the case for why you need a 3D structure when you're bioprinting. So based on this work, a company called Zonula reached out to us from Montreal, and they make an adherent antagonist, which was capable of inhibiting, I think, breast cancer cell adhesion. So just to quickly review, incadherin is a cell adhesion molecule that's commonly overexpressed in tumors. So you'll see it as the cells, how they adhere to each other. And the more incadherin you usually see, the worse the tumor is. So a wonderful graduate student, a former graduate student, now graduated, Emke Smets, who visited us from the Netherlands, and she actually just visited us recently as well. She decided to test this in our 3D bioprinted ring structure. And she found that this drug was pretty effective at killing off some of the cancer cells. And then she went ahead, and this is where the title of my talk comes from, about 3D bioprinting complex tissue models. So she actually printed these cancer cells alongside healthy human astrocytes. And they're in rings again, as you see here. The reason they're pink is because our ink is normally clear, and if you print it pink, then you'll know when it actually clogs. And then she did something really smart, was she decided to actually culture these tumor models for 16 days before adding the compound. So they actually were able to form the tumors before treatment. And again, normally in real life, when you're detecting brain cancer, it's after the tumors have already formed. She also used, she did not label the astrocytes, but she did label the glioblastoma cells. So you can see them right here. So you can see day 16, when none of the groups had been treated, everything had really large tumors. But then at day 21, you can see that the groups that had been treated with the higher doses of drug had significantly less GFP. And then by day 30, all the groups that were receiving treatment had significantly less GFP expression. EMCA went ahead and quantified all this. So you can see statistical differences in the huge decrease in GFP at day 22, and then for all the groups at day 30. And if you want to read more, this novel incadherent antagonist causes glioblastoma cell death in a 3D bioprinted co-culture model. It came out in Biochemical and Biophysical Research in the summer of 2020. This journal is really interesting because when you submit your paper, they say within a week, they'll either be accepted or rejected. And luckily for us, ours was accepted quite quickly. It was also nice because this was in the middle of the pandemic and we didn't have access to my research lab. So yeah, we were really excited that we were able to print this complex model of glioblastoma cells in the presence of healthy astrocytes, and that we identified a novel compound that was toxic to glioblastoma and could shrink tumor spheroids. And now, quite excitingly, we are actually replicating this work on patient-derived cell lines that we received from a collaborator at UCLA. Okay, and then one other sort of interesting story to just show you the sort of wide range of different technologies out there. We collaborate with Dr. Ryan Flanagan's group, who is at the Vancouver Prostate Center, and he evaluates the genetic and molecular mechanisms that contribute to non-obstructive azoospermia when patients are unable to produce sperm. And so we collaborated to 3D bioprint testicular tubules for sperm production. And so this was quite interesting because in this case, for certain patients, the problem isn't that they don't have the right cells to produce the sperm, it's that the structure internally isn't properly formed. So in this case, what he did is he actually took a patient's biopsy, obtained that patient's sperm stem cells. Megan Robinson, who works as his lab manager, expanded out millions of these cells, and then we printed them into testicular tubule structures. So this field, compared to neural tissue engineering, is relatively understudied in terms of there's so much biology that's known about the nervous system, how to make these cells from iPSCs, and so forth. And so we were really trying to see what would happen if we were able to bioprint these adult testicular cells into the proper structure using Bioprinter, and then compare them to the current gold standard for this, which is a 3D testicular organoid. And so using these patient biopsies, we again got personalized cells, and then we used our 3D bioprinter in order to replicate a testicular microenvironment. So this is where we actually did use the 3D printing system. We used a coaxial flow printhead. So in this case, we were actually printing these testicular tubules that had a core shell morphology. So it had an external core and an internal core, and this is the structure that we were trying to mimic from an in vivo setting. And the cells are pretty happy in it. So again, Megan was printing at 1 million cells per mil, and the majority of them were alive, as you can see here. Interestingly, when we compared these to the organoids, we actually saw the 3D bioprinted testicular tubules expressed more of the genes associated with spermatogenesis compared to the traditional organoids. So this was quite exciting, showing that potentially by having the structure, we were getting better differentiation into sperm, at least early sperm, by using this structure compared to having just an organoid present. And this really becomes clear when you look at the histology, where you can really see this core shell morphology of both of them compared to sort of these little organoids that are little round balls. And so being able to actually use bioprinting to make this structure, and again, these are some of the genes that are expressed in the testicular tubules, and then this is just H&E staining here. But you can really see the core shell structure and how that really has an influence on maturing these cells as well. And as I mentioned previously, if you're really looking to get into doing some of our, to do some bioprinting, we have a really nice STAR protocols paper that's got all the details you need on how to do and implement this. It does talk about both, as I said, the cheaper alginate gelatin. It also talks about how to print into BALs, which allows some more complexity as well. So yeah, and again, this is also open access, so if you are wanting to just go out there and look. And as I mentioned, we have both the cheaper ink, which I recommend playing around with, and then if you want to get into printing human tissue models, you can call Axolotl, and we'll send you some of our bio. Yeah, just with last couple of minutes here, I just wanted to talk a little bit about Axolotl and founding a company in a pandemic. I was always one of those people who was never going to found a company. So of course I did, and of course in the worst time possible during a pandemic. So we spun off Axolotl in March 2020. We had known we were going to spin off the company since the previous fall, and we had been working internally with one of the innovation centers at the University of Victoria. They helped us do our incorporation. We had won some internal money and some internal competitions to bring in funding for this. And we were quite lucky that we had received government funding. So when we incorporated, we initially had 5 employees. And in fact, of those initial 5, 4 of them are still with us to this day. And the 5th, Misha, is finishing up her degree. I co-founded the company with my PhD student, Dr. Laura De La Vega, who minored in entrepreneurship, and she was quite excited about it. And Lila Abelseth, who also was one of the people who you probably remember seeing Abelseth names on a lot of the papers associated with tissue print. So that was our founding team. Kaylee has been there since the beginning. She previously worked in the Willerth lab, and now she's the production manager at Axolotl. Alex is our business analyst, and she's great. We just spent today doing a bunch of calls together for Axolotl things. We're currently going to be doing the Creative Destruction Labs program, so we're pretty excited about that. Dimitri's our e-commerce specialist, but he's also great at all things 3D bioprinting. Salim is a recent graduate who does our quality assurance. Melina is our superstar biomedical engineer. I think you probably saw her names on some of those papers. She also has a paper out there where we bioprinted mesenchymal stem cells. And I currently have a paper from her that she wrote. She's working on our 3rd product, a bioink called Heartprint. Tara's our production assistant, and Anna Dunphy's our biomedical engineer, and Josh is our co-op. And if you are interested, we have lots of really great social media, thanks to Anna. We're on Twitter, LinkedIn, and our Instagram's pretty cute as well. With that, obviously nothing gets done without our funding agencies, as well as our current and former lab members. I think I've highlighted most of the people who've helped out. And yeah, with that, I'm gonna stop sharing and hopefully be able to take some of your questions.
Thank you, Dr. Willerth, for an informative session. I would now request the participants, if you have any questions, please type in the comment sections, or you can raise your hands before unmuting your mic.
Let's see. Okay. Under show participants. Okay, I think it's Shanzea. Do you want to ask your question with your hand raised?
Okay, going.
Hello. I don't think I actually have access to the chat, but maybe if you could type it in the chat and someone else could read it, since it sounded like there was a lot of feedback.
Yes, Dr. Shopee, I will read the questions from the chat. Shanzib,
Okay.
if you have any questions, please type in the comment section. Okay, Dr. Stephanie, we have a question. What practical tips would you like to give to the participants to start bioprinting in Pakistan?
Yeah, I mean, I would say it would probably depend on how much money you have, but like even before it took forever to get our aspect system, and so we actually did a lot of our ink optimization with microfluidic pumps. And as I said, there are ways to home build your bioprinters. So I would probably start by looking for some of those methods. I think Adam Feinberg's group has, they have a paper published on this, and actually they have a workshop every year that people can attend, and it's I think like $2,000, and you'll actually build the bioprinter at Carnegie Mellon in Pennsylvania, and then you get to take it home. So that's probably where I would start. And I'd also, as I said, check out our Star Protocols paper to see some of the cheap inks, because it does take a while to like get used to the printing process, and you don't want to culture a bunch of cells like we do and then print a bunch of them, just have them die. So that's why I always recommend doing a lot of test prints as well. And yeah, you can also see, I know it's a little bit different, but I do think, yeah, I think they're pretty cost, like Selling has pretty cost-effective bioprinters for their BioX and things like that. There might be some Japan companies as well, but you could also always try to see if you can get someone to come do a demo with the different types of bioprinters. So that's what I would suggest.
Okay, Dr. Stephanie, we have questions. What can Mueller engineering do in degenerative diseases like encephalitis and Parkinson's?
Sorry, can you repeat that?
What can Mueller engineering do in degenerative diseases like encephalitis and Parkinson's?
Yeah, well, I think we've been pretty excited. I didn't talk about it, but we have been bioprinting some Parkinson's models, and you do see Lewy bodies. So now that we actually have them, the idea would be to actually treat them with drugs to see if we can prevent the formation of those structures in vitro. And then if it looks promising, we can translate them for in vivo applications. Our Alzheimer's work is a little bit more direct because we are actually printing the patient's own cells. So if we do find a drug that reverses symptoms in that patient, Hackam could potentially prescribe it directly back to those the people whose cells we're actually analyzing. So I think that that's really, really powerful, is the use of being able to like have a patient-derived iPSC line with disease phenotype and actually being able to connect that back to the patient. I think is going to be one of the big game changers. And the fact that with our technology, we've been able to keep our cells alive and our models working really well.
Okay, so we have a question. Is some work also being done on artificially producing ovums in case of infertility?
Yeah. So as I mentioned, Ryan Flanagan's group, they do print the testicular tubules, and while we're only making immature sperm now, if they were to make mature sperm from that patient's cells, those could be used for IVF. There's a similar group working on making eggs that's out of Italy as well. So a lot of interesting work going on there.
Okay, Dr. Stephanie, what has influenced you the most to do this sort of research in tissue engineering?
I've always been fascinated by stem cells. When I was in high school, I actually wrote my senior thesis on stem cells. I'm old, so that was the cloning of Dolly, which I was just infinitely fascinated by. And then when I was a graduate student is when I was actually at the meeting where Yamanaka announced the discovery of iPSCs, which is funny because at the time I was like, this feels like this should be a much bigger deal than it is, and it was. So yeah, I just really think stem cells are fascinating and their ability to sort of replicate lots of different types of tissues.
Okay, Dr. Stephanie, we have a question. How we can minimize the risk of contamination in 3D bioprinting?
Yeah, so we actually use cell lockers that are filtered because we actually have in our lab the bioprinters in a different cell culture room compared to where we normally culture our cells. We actually we bought lockers from, I think, Thermo Fisher. So essentially they're like mini incubators that have the same filtration to keep things sterile. We also bioprint in an actual hood. I know a lot of people don't, but we do actually print in a sterile environment. And I think a lot of the newer bioprinters have built-in sterility, which is nice. But I do know for so essentially we're really careful. And also we used to, even pre-COVID, people in my lab wear masks because we're actually working very closely with the bioprinter because you have a lot of the hands going in and it's sort of like right at breathing level. So yeah, we just try to be really careful, follow sterile technique, and use cell lockers and things like that.
Okay, we have one more question. Can the testicle tubular printing be successful procedure for intersex people?
I think you could. I know that we've had some interest from like we have a chair on transgender studies at UVic, and he was really interested in the fact that you could potentially do that to treat people. And actually, a lot of the people who've worked with Megan's group with the prostate center are actually sort of in that kind of group as well. So yeah, I think that there's a lot of potential there. And I think, I know that that's something that my collaborator Ryan's really passionate about, is, you know, helping infertile couples to have children.
Okay, thank you, Dr. Willerth, for this informative session. I would like to present certificate of appreciation to our honorable guest speaker, Dr. Willerth, on behalf of Arts and Literature Society, Salim Habib University.
Oh, thanks. Well, it's great. And then thanks for having me. It's really nice to see so much interest in the field, and hi from the other side of the world.
My screen is visible. The certificate is visible to you.
Yes, we can see it.
Okay. Now I would like to request Ms. Sanya. Now I would like to request Ms. Sanya, student of Pharmacy Department and President Arts and Literature Society, to say a few words for our honorable guest speaker.
Hi, Stephanie. This is Sanya Ahmed, the President of Arts and Literature Society. It seems like everyone I talk to wants me to appreciate appreciation for your words and teachings throughout this entire seminar. A few of our members recently had a very good fortune of going through your works, and we thoroughly enjoyed them. Thank you for speaking to us today and extending your knowledge with us. We are grateful for the time and effort you took to share your thoughts and experiences with us. I believe we can benefit immediately from the incredible advice and suggestions that you have provided. Please consider adding our group to our annual speaking tour. We really, really enjoyed it. You are welcome at our conferences. Thank you for being a part of the session.
Thanks so much. Thanks for all your kind words.
Okay, thank you, Dr. Willerth, for giving our valuable time to us. Thank you, everyone, for attending the session. Thank you, Dr. Stephanie. Have a great day. Allah Hafiz.